So now we have seen how the 48s open complex is formed. So this is our 40s subunit. This is mRNA. Here is the start codon. Here are the anticodons which are bound with the start codon at the P site. This site is actually P site where this whole thing is occurring, that is ternary complex is binding with the start codon. This is the P site. Here is our initiation factor 5, 3, 1 and 1A. This is our 4F initiation factor, and here is the initiation factor 2 in the phosphorylated form. So this is our structure of 48s open complex. Open complex. Now what happens? Now these factor 5, 3 and 1, they get removed. There is breakdown of GTP to GDP so that in the dephosphorylated form factor 2 also gets removed. So all these have been removed now. Factor 5, 3, 1 and dephosphorylated factor 2. They all get removed, but factor 5b in the phosphorylated form gets attached. The main function of this factor is that it helps in binding 40s subunit with the 60s subunit. So 60s subunit comes here. Factor 5b phosphorylated also comes, and this factor helps in joining these two, that is 40s subunit with the 60s subunit. So here is our phosphorylated factor 5b. All the factors, that is 5, 3 and 1 gets removed. Only factor 1a is present here. So this whole structure, that is 40s subunit with the 60s subunit with the initiation factor 5b phosphorylated, initiation factor 1a and initiation factor 4f attached to the cap. This whole structure gets synthesized and it's known as the 80s initiation complex. After this initiation complex formation, we have to convert this complex into the elongation complex. For that, all three gets removed, that is 5F also gets removed, 1A also gets removed and 5B also gets removed after dephosphorylation or after conversion of GTP to GDP. So all three get removed. This is factor 4F, this is 1A, this is dephosphorylated 5B. So that we actually are left with this smaller subunit with larger subunit with our mRNA here, and no initiation factor is present. All have been removed and this complex gets changed into the elongation complex. Now, so this is our initiation factor 2 dephosphorylated. It combines with initiation factor 2b and they form a structure like this, that is they gets combined with each other. When this combination is formed, GTP gets converted into GDP and this factor initiation factor 2 gets phosphorylated. So after getting phosphorylated, they break into the separate subunits, that is phosphorylated factor 2 and the initiation factor 2b. So formation of this complex is essential for the conversion of GTP to GDP so that this dephosphorylated factor 2 gets phosphorylated. So when it gets phosphorylated, this complex breaks into phosphorylated factor 2 and initiation factor 2b. So this factor, that is phosphorylated initiation factor 2, again gets used in the formation of ternary complex. So let's see the whole process again. This is our 48s open complex. Now all these initiation factor gets removed, that is factor 5, 3 and 1, only initiation factor 1A is left. This factor gets dephosphorylated with the use of energy and gets removed in the dephosphorylated forms. So here is our initiation factor 1A. One more initiation factor gets attached, that is 5B phosphorylated. This is needed for combining these two subunits, that is 40S with 60S. Our initiation factor 4F was always present here attached to the cap. So this whole structure is known as initiation complex 80S. When all these initiation factors are removed, that is 4F, 1A as well as 5B, then this initiation complex gets changed into a elongation complex.